So we are about to begin. May I request all the members to please keep your mobile or laptop muted. Okay. Namaste and good evening all. Respected teachers, colleagues, dear friends and students, and distinguished speaker of today's webinar, Mr. Manoj M N, and the moderators of today's session, Mr. Harry Jose and Mr. Rudraksh. On behalf of CoPS Global, I extend a warm welcome to all the participants who have joined with us today for the third talk of the international webinar series, Pharmaphare, a guiding light for the next generation pharma professional. Before we begin, let us invoke the blessings of Almighty with a silent prayer. Thank you. This is our third webinar series, and majority of those joined us today evening are aware of the objectives of this endeavor. And for those who have joined us for the first time, let me brief about the CoPS Global. CoPS Global is a collective of all the pharma professionals graduated from College of Pharmaceutical Sciences, Government Medical College, Thiruvananthapuram. We have joined here to create a common platform to share the experiences, Am I audible? Prashant, am I audible?
You are audible now.
Okay, thank you. Today's talk is delivered by Mr. Manoj M N, Research Team Lead, Bigtec Labs, Bangalore. He will be taking us through the details of molecular diagnostics in healthcare. We are indeed fortunate to have this highly experienced professional working in the development of diagnostics, an area where a very few pharmacy professionals enter. On behalf of all the participants here, I extend a hearty welcome to Mr. Manoj M N.
Thank you.
I also extend a warm welcome to the moderators of today's talk, Mr. Harry Jose and Mr. Rudraksh.
Thank you.
We're also expecting Dr. Bijoy Matthew to join us. He's traveling right now, and I hope he will be a part of this talk. I extend a hearty welcome to Dr. Bijoy Matthew. I also extend a warm welcome to all the students, teachers, and all pharmacy professionals who have joined us today evening for this webinar. Before moving on to the talk, we are back again, yet on another day of national importance. Today is November 14th, the birthday of Pandit Jawaharlal Nehru, celebrated throughout India as Children's Day. Pandit Jawaharlal Nehru, our first Prime Minister, gave immense importance to education. Panditji used to say that children of today will make the India of tomorrow. The way we bring them up will determine the future of our country. It was his vision that led to the establishment of pioneer institutions like AIMS, IIT, IIM, etc. And we are never too old to wish each other on Children's Day. It's just a sweet way of celebrating the innocence we all have in our heart. So on behalf of all the members of CoPS Global, wishing you all a happy Children's Day. In a way, we CoPS Global are also in the path of fulfilling the dreams of Pandit Jawaharlal Nehru. We hope to be a guiding light for our students for a better tomorrow, and in that way, we are indeed proud to be a part of nation building. Dear students, as we used to say before, this webinar series is designed with you in our mind. Feel free to ask any queries. Questions are always welcome. Please make use of the chat box. Please give your valuable feedback so that we understand your concern and can be of any help in your journey. We have received overwhelming responses from students and faculty from various corners, and we are happy to know that this webinar series is achieving its desired goal. Thank you all. So we will move on to the scientific session led by Mr. Manoj M N. He will be deliberating on the topic molecular diagnostics in healthcare. The session is moderated by two eminent alumnus of CoPS Trivandrum, Mr. Harry Jose and Mr. Rudraksharan. And also we are expecting Dr. Bijoy Matthew. Mr. Harry is a familiar face. He was with us as a panelist in our first talk of Mr. Samish Menon. Mr. Harry Jose did his B.Pharm from CoPS Trivandrum in 1999 and later on took his MBA in marketing from the prestigious Nirma Institute of Management, Ahmedabad. Harry is currently serving as the Associate Vice President and Global Head of Marketing at Syngene, Bangalore. Syngene is one of the leading global contract discovery, development, and manufacturing organizations in the pharmaceutical and biotechnology space. Welcome, Harry. And next is Mr. Rudrash.
Thank you.
Rudrash passed out B.Pharm from CoPS Trivandrum in 1996 and is presently heading the marketing at Idorsia Pharmaceuticals in New Jersey, USA. Idorsia Pharmaceuticals is a Swiss-based biotech company, a high-potential biopharmaceutical company specialized in the discovery, development, and commercialization of innovative small molecules. Welcome, Mr. Rudrash. Now, next is Dr. Bijoy Matthew. He is the Associate Professor of Pharmaceutical Chemistry, Amarda School of Pharmacy, Ernakulam. He is a popular researcher among the pharmacy professionals in Kerala. He was recently listed among the top 2% scientists in the list prepared and published by Stanford University and elsewhere. So welcome, Bijoy Matthew. So now we'll start the webinar. Wishing you all a highly informative session. That's it from my side. Over to you, Harry.
Thank you so much, Ranjesh. Before I start, I am audible, right?
Yes,
Yes.
Thank you. Thank you. So good evening, good afternoon, good morning, depending on which part of the world you are joining us from. I'm told that we have people from Europe and US, apart from India, participating in this event. So we are back with another very, very interesting session. This is the third of our Pharmaphare series, and it's on molecular diagnostics in healthcare. To those of you who are familiar about this program, I don't need to explain much, but in case there are a few people who are joining fresh, there is something very interesting that I want you to focus on with regard to this, not just this webinar, but this webinar series itself. All of the sessions in this series are done by your seniors, people who have passed out of College of Pharmaceutical Sciences, either in B.Pharm or in M.Pharm. So in some form, if you should really look at this as an alumni meet, where you have a chance to listen to people who have passed out from the same institute that you have you're studying in, and a chance to really ask some questions and take some guidance of the path that they have traveled. And hopefully it will give you some guidance about the opportunities which are available to us, the massive amount of opportunities which are available to us in the world of pharma and biotech. With that, let me just move on to today's topic and to today's presenter. Molecular diagnostics in healthcare is a topic which is of extreme prominence these days because of the COVID pandemic. But it is a topic which, unfortunately, not too many people would be very familiar with. And hence, it's really our luck that we have an eminent person, an eminent researcher to take us through this very, very interesting topic. You know, with regard to things like diagnostics and all, the last time a term really caught the attention of the public was during the AIDS pandemic, 15, 20, 25 years back, when suddenly the entire public suddenly became aware of a test called the ELISA test. These days, everybody, even if you are in, if you ask kids who are in the 4th standard, 5th standard, they will also tell you about the RT-PCR test. So RT-PCR test is, of course, a very, very important diagnostic test. It has a lot of other importance in the biotech industry as well. And that is definitely one area which Manoj is going to talk about quite a bit. But the world of molecular diagnostics is much, much faster than that. And to take us through that, I will invite Manoj M N, who is currently a researcher who is leading research at Bigtec Private Limited in Bangalore. Manoj, as Ranjish mentioned, is an alumnus of College of Pharmaceutical Sciences, Trivandrum, 1992 batch. Passing out of CoPS, he did his master's in bioprocess technology from UDCT Mumbai. And after that, he did about 8 years of scientific work in Vital Malia Scientific Research Foundation in Bangalore, where his area of work included nutraceuticals, statins, and biosimilars. And for the past 13 years, he has been associated with Bigtec Private Limited, which is a company which focuses on molecular diagnostics. And without taking too much more of your time, Manoj, I'll just pass it on to you to talk us and lead us through this wonderful world of molecular diagnostics in healthcare. Over to you, Manoj. Thank you.
Thank you, Harry. Thank you, Harry and Rudrash, Ranjish, for the introduction. So I hope we can begin today's session. I understand it's quite heavily raining in Kerala and they have issues with connections, etc. But I hope people can join as much as possible. And as Harry mentioned, this topic is little away from the core of pharmaceutical subjects. It's about diagnostics, but still connected to medical profession. So I would like to take everyone through that in detail. I would like to keep it as simple as possible for that people can understand it. And we'll give an overview of the whole technology. So in the session is separated into three sections. So we have section 1, wherein we will go through about the molecular diagnostics and some technological insights into PCR and techniques used in pharmaceutical relevance. And I would like to give a path in case somebody want to do a diagnostic development, what should be the path taken. All these sessions are aimed at students. So I'm going a little technical into these parts for those who would like to know in detail and how to connect whatever you're learning to what is actually happening in industry. Part 2 would be about 10 minutes where we will touch upon an in general path towards a product development that happens in an industrial setup. And there's a section 3 for you where we will have a virtual scenario of being in a product development setup. Let us go through that. So the first part is about the molecular diagnostics in general. And as you know, the term molecular diagnostics means you use a molecule as the target as a biomarker. And if you ask me, protein is a biomarker, it's a molecule. But by convention, molecular diagnostics talks about DNA or RNA as the biomarker. And there are different techniques to be used for that. And most important, most popular is PCR, polymerase chain reaction. And we have microarray technique that is for probing mRNA expression, etc. We have sequencing, which is most commonly known today as next generation sequencing, where you read out the sequences. And this is from all the sequencing that you come about the variants of COVID and mutations, etc. Another technique quite common is called FISH, fluorescence in situ hybridization, which is about mapping aberrations in chromosome. In this particular session, we'll focus on the first, that is the PCR part, which has many variants within that. We have the conventional PCR, real-time PCR, RT-PCR, LAMP, and the number goes on. I would like to focus more on the qPCR and RT-PCR because that is the buzzword today, as Harry has mentioned. Now, what is the source of this nucleic acid that you are analyzing? So most, it can come from two sources. One is the human genetic test, wherein the target nucleic acid is the DNA or mRNA or microinterfering RNA or circulating free DNA.
Manoj, can you just type the microphone? There's small vibrations coming from your microphone. Just
okay. Is it better now?
Yeah.
Hello.
It's okay. It's okay now.
Okay. Thank you. Thanks, Prashant. So this is about the human genetic test. Mostly go for oncology or any other genetic predispositions. But the majority of RT-PCR or PCR tests that are happening today is of some kind of infectious diagnostics. And if it is from a DNA pathogen, mostly a bacteria or some viruses, we call it PCR. And if the target is an RNA, like in COVID, it called for an RT-PCR. RT stands for reverse transcription. So what happens is that first we need to convert the RNA to DNA, complementary DNA, and then amplify. So it starts for a pre-step that is before the PCR. And as you can see, majority of the diagnostics is about infectious diagnostics. Now, coming back to the techniques of PCR, I hope you would have studied this from school and early in colleges and many places. And it's a chain reaction starting from a single target DNA, and you amplify that into 2 copies. That become 4 copies and 8 copies. And if you look at the equation, it is 2 power n, where n stands for the number of cycles, and you get an amplified product. So what happens in each step is little elaborated here. So if you look at a strand of DNA, the top strand and bottom strand. So when you heat this molecule to 95 degrees, it's separated into 2 strands. And we have some reagents in the reaction is called primers. These are nothing but small fragments of synthetic single strand DNA that anneal with the respective strands on top side or bottom side. So this is called forward primer and it's called reverse primer. And we were amplifying the region between this called as an amplicon. So when you do a PCR, the enzyme binds to these molecules and then extends the reverse primers in this way and the forward primer in this way. And the reaction goes on for every cycle, and you get 2, then 4, then 8 as it goes on. And at the end of the reaction, we take the content of the tube, PCR tube, and load it on a electrophoresis gel, and wherein the fragments get separated according to molecular weight, and we identify the fragment of interest against a standard. So suppose you are looking for a 100 base pair fragment, it should match with the 100 base pair on the ladder that we call it as the standard here. So this is conventional PCR. So you get the information only after the PCR and after running the gel that the test is positive or negative. Now let's see what's different in real-time PCR. Here we have another small DNA strand labeled with the 2 molecules on either end. One is a fluorescent molecule. We call it as FAM. This is a green fluorescent molecule. And on the other side, we have a quencher. It's very common to have a black hole quencher here. Okay. And this probe is called a FRET probe. FRET stands for fluorescence resonance energy transfer. What happens here is when you have this kind of a molecule, even though this compound is fluorescent, it cannot emit much of a light because whatever this is emitting will be absorbed by this quencher when it is in this distance that is called Foster radius. So even though this molecule is there in the solution, when you shine a blue light, it doesn't emit much of green light. But when this reaction is going on, this Stack polymerase has a property. Whenever it sees a molecule in between or in the path of that, it cuts these probes into pieces so that the reaction can proceed. When that happens, you have these now molecules separate, not connected by the oligo chain, and this moves much apart in the solution, and this FAM molecule now become active. So when you shine a blue light on that, this will emit a significant green light. And as the reaction goes on, for every reaction, we have this 1 molecule of FAM getting accumulated in the reaction. And at the end, when you illuminate the tube with a blue light, you can see it is fluorescing. So here, the advantage is that as the reaction going on, every cycle by cycle, you get a signal and it will indicate whether the reaction is going on positive or it is negative or is there any other issue with that. Hope that is the most simple explanation of what is a real-time PCR means. We will go into the technical insights into that in next slide. So if you have any questions, you can ask it We'll have a break between each section. So you can ask it there or you can ask it at the end of the session. I would like to explain if you need any more information on that. So let's look at what's happening in the tube. Now, this is at the molecular level. So let's say you have starting with thermal cycling, the normal thermal cycling between 60 degrees and 95 degrees. So it starts at 60 degrees, goes to 95, stays there for some time, few seconds, and comes back to 60, stays there for some time, and it repeats. And what you see in blue color is the number of cycles. And as I mentioned in the earlier slide, if you start with a single DNA, that becomes 2 DNA after first cycle, it becomes 4, 8, and it goes on. So I'm not showing all the cycles here. We have broken in between. Let us come to that. Now, every cycle at this 60 degrees, the reaction happens. It makes 2 DNA and it creates one fluorescent molecule free. And when you measure this intensity using a photodetector, you get a value there. You plot that. And you go to cycle number 2, now you have 2 molecules, you get some intensity. Not that it is not changing, I will come to that point later. And cycle 3, you have 8 molecules, you have some signal. But as the reaction goes on, by around 35 cycles, this number would have become a huge number, more than 1 crore molecules in solution. At that time, the intensity start to show up, slightly increasing. And as the cycle goes on, if you notice here the number 3.4, that becomes 6.8, doubling, and again doubling, doubling, doubling. But in this region, the intensity is much higher and much easy to measure. So we choose a base signal level on the y-axis, we call it as a threshold, and we calculate at which cycle, like here in 36 cycle, it has crossed the threshold. So you get this kind of a growth curve or X sigmoid curve, what people call by different name. What is most important is value called cycle threshold. That is at which cycle the signal has crossed the threshold. Here in this case is 36. Now that is what is happening in the tube at the molecular level, and this is the controls. We have thermal cycles, and then we have fluorescent intensity measurements as and when the reaction is going on. Now let's see one simulation. What happens if you see, this is the bottom graph shows the thermal cycling, temperature going up and down, and top graph now it is 0, but just watch out. The reaction is going on, you started to get increasing level of signal intensity. Now this is what you typically see. Now the reaction takes typically about 30 to 1 hour and depending on the machine. And as the reactions are going on, every cycle you get a signal, and initially it will be near 0. Then at particular cycle, you can start to see it growing exponentially. Now this video keep looping. What is more important is to watch out these numbers. How many DNAs are we starting with in each tube? In tube number 1, which is in blue color, we have 10 DNA molecules. Next tube we have 100. Next tube we have 1000. And if you watch out, 1000 starts early, followed by 100, followed by 10, and these are equally spaced between each of them. To be precise, that will be 3.32 CTs separated from each other if the DNA have a 10-fold dilution. Like if you starting with 1000, 100, and 10, these 3 curves will be spaced by a factor of 3.32 CTs. We'll come to that later. So essentially, this is what is happening in a real-time PCR. We have thermocycling going on, then we have fluorescent intensity, and you have signals amplifying, and the technician note down the value at which the reaction crosses this threshold signal. So once you get the threshold signal, how do we calculate? You know, there is a connection between the starting molecule and the cycle at which the intensity increases. But mathematically, how will you use that to back calculate how many DNA you started with? So that comes on this slide. Basically, show this, the meaning between the CT value and the starting DNA count. So let's see if you get a standard curve, like if you plot the CT value that you obtained in a serial dilution Here the DNA count is in logarithmic scale. So 8 means 10 power 8, then you have 10 power 7, 10 power 6, 10 power 5, 4, 3. You can see that this becomes a straight line having a unique slope and an intercept value. So that is advantage of plotting it in a log linear scale. So CT values on a linear scale and log of DNA on a logarithmic scale. So suppose you do a reaction and you got a CT value around, let's say 26. Now you can find out the corresponding DNA level on the x-axis. Let's say this is 4.3. Now what you do with that to the sample? So here, let's say you have started with about 500 microliter of a blood sample. Let's say you are analyzing hepatitis B DNA or something like that. You need to do some cleanup because blood has so much of inhibitors. It has hemoglobin, it has proteins, lipids, etc. So these things need to be cleaned out. We call it as a DNA preparation. And you will have about 100 microliter of elute. And that from that we take about 10 microliter of DNA elute into the PCR tube and do the PCR. From this PCR, we got a 26 CT, which is corresponding to 4.3 log of DNA. Now we need to convert this 4.3 into number. So what you do is that you take a 10 power 4.3, that gives you an absolute number, that is 19,000 copies in this 10 microliter. Now you can back calculate how much will be there in this 100 microliter because we have taken only a portion of that for PCR. So we'll have about 1.9 lakh copies. We generally call copies, it means number of counts in this 100 microliter, and all of them has come from 500. So back calculate how much would have been there in this 500 microliter, and that's about it's about 1.9 lakhs, which came from 500 microliter. And what about per ml? If you want to do per ml, multiply this by 2. So you get about 3.99 lakh DNA per ml of blood. This is how qPCR is used to quantify the pathogen load in the starting sample. So this is used widely for monitoring therapeutic, the treatment and then treatment progress in patients like hepatitis, HIV, etc., where medication called AR treatment is given. And every 3 months, blood sample is drawn from the patient, and we check for how much of DNA is there in the, or DNA or RNA there in the blood sample. And if it is decreasing significantly, that means the treatment is working and the patient can continue to on that. Or if it's staying same or it's increasing, that means the treatment is failed and you need to switch over to another medication. So that is how qPCR is used for monitoring a treatment progress. Other than that, for many diseases, this will tell you how severe is the infection. If you get a higher amount of starting viral load or pathogen load, that means the infection is at a higher level. Or if we start with a very low level infection, very low copy number, that is a mild or very insignificant level of infections. So in all these ways, that indicates how the severity of the infection is, how much is the load of pathogen, etc. Hope that is clear to you. Moving on, coming to other tests that we generally have, human genetic tests. These are mostly some kind of aberration in human DNA. Some of the couple of prominent information is on sickle cell, the sickle cell trait and thalassemia. These both are related to blood anomalies. And in sickle cell, if you look at, we can monitor the change in the human DNA. If you look at carefully, this is a one nucleotide change, which is called a single nucleotide polymorphism, or SNP in short. What happened is that in normal case, it is CTC encoding for glutamic acid. Is a one amino acid in the hemoglobin chain. But in sickle cell cases, that becomes CA. So T here has become A. That caused the amino acid change from glutamic acid to valine, and that created an abnormal hemoglobin chain or protein, which distorts the shape of RBC and its blood carrying oxygen carrying capacity. So any of these human DNA changes we can detect using PCR. So that is one application of PCR for human test. Thalassemia, on the other hand, has multiple mutations and according to the origin. Another example is in cancer treatment for leukemia monitoring. So cancer, acute myeloid leukemia or chronic myeloid leukemia is caused by a fusion protein called BCR-ABL. So this is a interchromosomal jumping gene which forms a fusion gene, and if you monitor the presence of this gene, you can see the patient is responding to treatment or not. Another now most popular one is monitoring chromosomal aberrations in fetus. It's called non-invasive prenatal test, wherein multiple parameters are tested to identify Down syndrome. Mostly these are multiple copies of chromosomes. Beyond 2, we have 3, like chromosome 18 in 3 copies or chromosome 21 in 3 copies, which is causing some kind of a genetic disease. Also, another example is autoimmune markers. For example, human leukocyte antigen. This is an antigen on white blood cells that is implicated in some kind of autoimmune diseases like spondylitis and reactive arthritis. So if you do a HLA-B27 test from your blood sample, you can find whether you have a predisposition to getting a chance for these diseases in that. And human, that's a big list for human genetic tests, giving some example to identify. Another topic of interest, especially in pharma, is called companion diagnostics. So companion diagnostics are diagnostics that is co-developed along with a therapeutic agent. What it does is that it identifies people who can have a positive response to that particular medication. So these are important for long-term treatments like cancer treatments or psychiatric medications, etc. So these IVDs provide essential information whether the population that you're treating will respond to that particular medication or not. So it is very much tied to that particular treatment. So let's take an example of a cancer treatment. So you have a normal population, you do a diagnostic test, you identify a few people as the positive for the cancer test. But these have people who may respond positively or not respond to the particular medications. So when you do a companion diagnostic test, you can segregate the population into 2, and you only need to treat this group with that medication. There's no point of treating this group with that particular medications. That's also important sometime on the drug side effect severity, because some population may be more prone to side effects and some population not. So this started very much in 1998 for a breast cancer treatment, a medication called Herceptin. It's a monoclonal antibody with this name. And along with that, they made a test called Hercept test. Basically, what this test is, it checks for overexpression of this receptor molecule called HER2. And only in patient population where HER2 is elevated, this medication is effective. So if you have a breast cancer patient and HER2 is level normal or below normal, there's no point of treating with Herceptin. So that is the idea of companion diagnostics. And if you go to the FDA website, there is a big list of cleared companion diagnostics, and I think there is more than 44 tests at present in FDA approved list. A little more extension to this is pharmacogenetics or pharmacogenomics. So this is more about identifying drug metabolism and then that changes in different population or different patients. So these are mostly used for long duration treatment medications like people taking blood thinner, or people taking some kind of antidepressant, or cancer patients, or psychotic medications, wherein the patient is taking a medication for a long time. And what you're monitoring primarily is the cytochrome P450 enzyme changes in those populations. Like if we have people who metabolize certain medication fast because of certain mutations in these proteins, or we have people who may not metabolize it that fast, causing the drug to accumulate in the body. So whenever we have a long duration treatment, it's good to know what is the genetic profile of the patient so that we can adjust the dose accordingly. When it comes to identifying a single protein, we call it as pharmacogenetics. And when it comes to identifying a group of proteins, we call it pharmacogenomics. So for example, let me pull out a report. This is from a company called GeneSight. And when you have a psychiatric treatment, we first do a gene site profiling of C450 activity, whether they are normal, ultra rapid metabolizing people, poor metabolizing people, etc. Accordingly, they will give an advice on what medications can be chosen for that patients and how much the dose can be adjusted. So this is a very important factor for pharma related field, where you call personalized medication. Like, it is not the common one dose for everybody. So the dose is adjusted, or the medicine is chosen according to the genetic makeup of the patient. With that, now we have an idea of various kind of molecular diagnostics. We have human, we have infectious. Infectious is so common, so we have like tests for dengue, malaria, chikungunya, to hepatitis or COVID. I'm not talking about that because that is so common nowadays. Now let's come to a typical path. How you develop this kind of test. So this is something important for students to know. So here I would like to give you an insight into the typical course on how do we develop a particular diagnostic for an application. So it starts with in silico design. This is mostly what you call bioinformatics, and wherein we design the primers and the probe. This is called the Tachman probe, where that gives the fluorescent signal out. So 3 single stranded oligos are synthesized based on the target DNA or RNA that you want to sense from the sample. And what is underlying principle in this design is your thermodynamic parameters. So thermodynamics is not just related to steam engine or other parameters. It is very much involved in biological calculations. And if any one of you is looking for a computer-aided drug design or drug screening, then again you will see this delta H, delta G, and those values. And this plays a larger role in designing and selecting the reagents. Next step after the initial design is developing a formulation. This is called the master mix, wherein we add the other reagents, the biological reagents, the enzymes and buffers and small molecules, etc. So when you do this formulation development, this is mostly we are playing with number of molecules, molarity. I hope all of you have studied these things, but this is largely put into use in this kind of formulation development, kinetics of enzymes, because PCR is an enzyme-based reactions. And as with any other enzyme, we have an enzyme kinetics and we have enzyme efficiencies to be monitored and to be maximized for a better reactions. So we have calculations based on number of molecules, molarity, and kinetics in this. That part of the formulation development is largely a biology chemistry domain interaction. And in all these due course, we have technical documentation, especially if it is an ISO 13485. So people with good skills in technical writing takes care of their job, and there are formalities to be kept, design dossiers to be made, design history files to be retained, because these are all subject to audit on a later period of time. It's not just important that I made an assay, it's always important to show the regulatory team that the path that is taken during the course of development, that there is no pitfalls in that development. Once the assay is kind of formulated, then we do a set of testing to know the analytical performance. This is where we identify the performance metrics, that is kind of limit of detection, lower limit of detection, upper limit of detection, linear range of the assay, the precision, the accuracy, all those parameters. And there are some guidelines internationally. These are called CLSI guidelines. This stands for Clinical Laboratory Standards Institute. So basically, the idea is that if every manufacturer should follow same guidelines, so these parameters or the technical parameters are expressed from a uniform set of experiments. So there is not much change from manufacturer A to manufacturer B, and everybody follows uniform guidelines. So this data is also submitted for approval by regulatory agencies. In India, it is CDSCO. In US, it is FDA. In Europe, it is CIVD, etc., or WHO. So in this, a particular area, what is more important, we have a lot of data analysis and statistical parameters, because all these values are expressed in terms of some kind of a statistical number. You have standard deviations, you have coefficient of variations, etc. Then once the assay is developed and the analytical performance is satisfactory, we get into the clinical performance. This is where we start testing the actual clinical samples, and clinical samples are diverse and samples from different patients behave differently. So the significant number of sample size is decided, and it is done in collaboration with other national laboratories or hospitals, etc., wherein we make a study plan, calculate the number of samples to be tested. Protocol is developed and approved by a competent agency, like somebody like ICMR or something like that. And the study is performed and results is analyzed again. Mostly it's a statistical analysis. Here we typically use a 2x2 matrix. You may call it as a contingency table or a 2x2 table, where we identify the clinical sensitivity, clinical specificity, positive predictive value, negative predictive value, kappa, and confidence interval, etc., etc. This is largely again statistics that comes into play when you analyze this clinical data. Once you have the clinical performance is also okay, we apply for the regulatory path. And in India, path is called CDSCO. We apply for a test license. And once you obtain a test license, we need to create certain lots of this test in the manufacturing setup. So this part of the initial study happens from the R&D, and from here the technology is transferred to the manufacturing unit, and according to the SOP, they create test lots. And again, it goes for another round of clinical evaluation, and if the performance is satisfactory, obtain a manufacturing license from CDUC again and start manufacturing, and it goes to market. And once the product in the market, it is very important to monitor how it is performing. We call it a post-market surveillance, and it's part of ISO 13485. And there again, it is data analysis and visualization, but in this case, it is going to be big data. So we have millions of data coming in to be analyzed, and then we need to monitor how the test is performing. Is there any aberrations, any issues, and it is to be notified to these agencies as and when that is happening. So CDSCO in India, it is called the Central Drugs Standards Control Organization, and that controls drugs as well as medical diagnostics. It is part of Drug Control General of India. And if you look at the medical diagnostic, there is medical devices and in vitro diagnostics in that. And we particularly come into this segment of in vitro diagnostics. So that's about India part. But if you look at outside India, if you're looking at European regulatory, it is called CEIVD. And if it is in the US, it is called US FDA. Or if it is in Asian countries in general, we call it's WHO pre-qualification. Like if you want to take a test from India to multiple countries, it is better to have a WHO approval. So that's in a nutshell about the development of diagnostics. I think I'm coming to an end of session 1. Maybe I have one more slide. Okay. So this is something I need to tell to the students. It's like when you're studying a subject, it is very important or very useful to use certain tools. These are tools mostly on data information, where you can work with databases. So you know about NCBI, and there are some other databases that give you specific information called Viper, Flu database, etc. And if you're looking for any of the COVID sequences, it's all in one database called GISAID. And I think there is more than 51 lakh genomes, whole virus genomes in the website, and maybe about more than 3.5 lakhs from India itself. So that's a huge repository on showing the genetic variation of COVID viruses. And from that analysis only, we are coming up with all these variants and mutations, etc., alpha, beta, gamma, delta, whatever we call. And another important database, if you are looking at biochemical pathways, this is called Coyote Encyclopedia of Genes and Genomics. This is a fantastic pathway It has everything from your glycolytic pathway to citric acid cycles and almost every drug metabolic pathway as well, which showing the metabolic enzymes involved and which is your starting material, which is end product, etc., etc. And if you look for any metabolic diseases, is a very important path to start with. Okay. And what are the design tools? So this set of design tool I'm talking primarily from a diagnostic point of view. This is to analyze the DNA sequence or RNA sequence. You have BioAudit, Primer 3 from MIT, and Oligo Explorer Analyzer to help. This will help you design the primers and probes. But if you're from a pharma background, you can use any of the drug discovery tools like CAD, tools for docking or molecular dynamics tools, etc., etc. And another important tool I believe every student should nowadays familiar is on analytics, because this is the buzzword today. Data analysis or big data analytics is the key point today on every domain, whether you are interested in marketing or sales, manufacturing or R&D or teaching. This is an important point for everybody to know. And we have very good, very easy tools to do this thing. All of these are free. You can use Python, you can use Pandas, all these runs on Python based systems. And if you ask me, Python is the most preferred, most loved programming software for any scientist and engineer. Now, why is that not C or Java? Because the amount of knowledge that you need in programming or the amount of effort that you need to put for programming is very less compared to the results that you get out of this program. You can do an entire data analysis with maybe as simple as 5 lines or 10 lines of code, and you can handle data in millions or even gigabyte range. So if you take a data in Excel sheet, I think old Excel has 66,000 rows. Now the Excel has 10 lakh data. But what if your data is more than 10 lakh? You cannot load into an Excel and analyze it. Whereas in Python or in using Pandas, you can have 1 GB or up to 1 GB of data, and you can even analyze that using a laptop. That is the beauty of these tools. And you can create stunning visualization. If you want to create graph, for teaching or publications, etc., there is a huge collection of templates to create visualization. So I would recommend every student, even in the first year of BPharm, to start familiar with these things. This will help you a long way, not only in your academics, but when you come to an industry. Now, why these tools? If you look at our evolution, the humanity evolved when we started using different kind of tools, right? It is from the Stone Age to different tools. And from line 1 to line 3, we're still using tools, but our tools has changed. We are starting with computers, mobile phones, and networking, and big data, and etc., etc. So why I have given these tools here is that all of these are free and open source. None of these need a paid membership, and every student can take a login ID or password, and you can access this information. You can use this freely, and you can learn all these things. Another advantage is not just learning these tools, it helps you better understand something that you're studying. For example, if you're studying statistics and you want to know how those parameters are being applied to data analysis. Now, when you're studying, mostly you are looking at the equations. If you ask me what are the equations for standard deviations, you give me equations. Equation for variance or coefficient of variance and another equations. But in application, that is not what is important. In application is how do we use what method to analyze, and what is the significance of the value that you get out of the analysis. Like, for example, let's say you got a standard deviation of 0.3. Now, what is the significance of 0.3 versus 0.6? That is a question when you come to actual data analysis, and if you're able to explain, and when to use mean, when to use median, or if you have studied all these things, you would know that these may give similar answers, but in some cases use median, some cases use mean, etc., etc. And the very easy way to understand all these things is to get hands-on with these tools and have a mimic database, and very many free databases online. Once you start using that, you get a very good insight on what tool to use and how to use it and how to interpret the results. That is the most important part of data analysis. Data analysis is not just running a software and getting a bunch of numbers out. How do you analyze, how do you signify that results output and present to somebody for better explanations? So that with that, Hari, I think I'm coming to the end of section number 1, where we talked about the qPCR, and in general, we've gone through the technique of PCR and other aspects, brushed upon the PCR aspects, other things. Now, this section number 2 is about, in general, methodology or towards a product development. Hari, is there any questions or something we need to take now?
Yeah, there are 2-3 questions, Manoj. I mean, mostly generic, not specific to this particular section. And for the participants who are listening into this program, we would suggest if you can just write your questions in the chat window. It would help us to pass it on to Manoj, and then he can answer. So maybe we can take a couple of those questions right now, Manoj, before we move on to the second section. So one of the questions which came up was, for a B.Pharm student, where really is the opportunity? What is the next step if they want to get into molecular diagnostics?
Okay, so I think I can best explain through using my career path.
Absolutely.
So,
that was the second question, by the way.
If you, okay. So I have taken, say after the B.Pharm, I took GATE exam and then got into biotech course. It's a MTech or MSc Tech in Bioprocess Technology in Mumbai University. And that specializes on bioprocess engineering and bioprocess technology. And then I could get into biotech R&D and eventually, based on the experience, etc., moved on to this diagnostics. So diagnostics is primarily, it's more of biology and chemistry. And I think we have enough chemistry and biology in B.Pharm itself. And what helped me more than that is an understanding of engineering subjects on pharmaceutical engineering and other related topics. So it's a bit of biology specialization after B.Pharm that can help in getting into these domains. And B.Pharm is a very diverse curriculum, and you can diversify into various things after B.Pharm.
Right. Thanks a lot for that, Manoj. So the second question which was there was on the market size, the India market, global market.
Yeah.
And what are the activities? What are the startups which are active in this? Are there any startups which is specifically looking at the diagnostic industry?
Correct. So if you would have seen the last year's growth, it was phenomenal, and it's like a balloon blown up for diagnostics. And other than that, the whole molecular diagnostic market stands at around $12 to $14 billion. It's a very quite sizable market, an emerging segment, though it is primarily centered in US. Asia Pacific is the most driving market, which is a very fast growing market as of now. And total IVD market in the globally is about $60 to $100 billion. And that is less compared to pharma, which is about 1 trillion. But these segments are increasing quite rapidly nowadays. And with these pandemics, now RT-PCR becoming a buzzword, and a lot of companies, major international players are being advised by their market analysis segments to invest on molecular diagnostic, because they see this as the major factors coming up. And there are a lot of startups in India itself. We have a lot of startups coming with innovative ideas on molecular diagnostics. Like we are one of the example, and there are many more. So opportunities in the field is rapidly growing. Pandemics is affecting somebody badly. On the same hand, it is creating opportunities for a lot of industries and job opportunities as well.
And Manoj, this is Rudraksh. How is it impacting the companion diagnostics industry? I think you brought up a very good point for pharma, especially.
Correct.
Right. So how is that evolving? Any scope in India for that?
Yeah, so that is definitely a growing market, and that comes along with these major pharmaceutical or therapeutics that is used for oncology. And oncology is a growing segment, oncology and psychiatry especially. And see, this is co-developed along with the therapeutic agents. So if you ask me, I don't know how much of therapeutic inventions happen in India. So these are mostly driven as of now by overseas companies, and if any company in India is doing therapeutics, then definitely that is being used. But nonetheless, many times these developments are offshore to India. Like if a company is developing therapeutic elsewhere, there's a lot of opportunity for Indian IVD company to make a companion diagnostics and support for them. So we were in part fought for a one major pharma major in and then supported them with the companion diagnostic.
Rudraksh, I think one of the other interesting aspects is that, along with companion diagnostics, one of the key things which is happening is this whole thing about data, patient data, which is now starting to get collected and being available. So once that happens, self-diagnosis and a whole lot of other things will also start to come into play. And at that point in time, the need for diagnostics is going to again exponentially increase.
Yes,
So
that's a great point, Harry. And
Yeah.
and in fact, as Manoj was talking, and Harry, you just bring up such a great point, and the whole space of like the self-COVID test. That was the other question that I see on the panel. That how does that technology really work, Manoj? The whole idea about the
Yeah.
self-COVID, for example.
Yeah, that has come off late to the market now. And see, there are two kind of, I would largely classify COVID test into two. One is an antigen-based test, which is a protein marker, and you have RT-PCR, which is looking at the genetic material. So all these self-diagnostic tests is more like a pregnancy test platform on the same lines. We call it as lateral flow technique, where we check for the presence of a COVID protein in your nasal swab, etc. So you have antibodies, and then you have a reaction cassette wherein the antibody is bound to the antigen if it is present, and then it illuminates a color, and you get a test line and control line. So this is in general the very old techniques like in the pregnancy test, the same platform that is getting applied to the self-testing category. But on the other hand, RT-PCR is more of a lab technique because we need machines and you need this test. And but the advantage is that an RT-PCR is many-fold sensitive than an antigen test. So that comes the trade-off. On the one hand, you can do an antigen test like self-test at home, and you can do an RT-PCR in the lab, but there is a difference in sensitivity.
Yeah.
Right. Yeah, so there was one other question which was there, which was about the latest regulations from the CDSCO regarding in vitro diagnostic kits.
Okay.
So is there anything, is there any new regulation? This question came from Saju Rajapan.
See, in vitro diagnostics is now, India came to the regulatory very late, on this particular domain, because we never had too much of diagnostic developments within India. Mostly we were relying on imported test kits. So regulation was mostly on regulating the trade and then efficacy testing in India. And once CDSCO incorporated a few years back, they started regulating IVDs in India strongly. And these are largely following the footsteps done in FDA and IVD. And if you look at those definitions are much more stringent and well defined, and India is evolving. But there is a very clear guidelines on if you go to the CDSCO site, it is very clear on what are the regulations being imposed, and it is becoming tighter and tighter, and to enable to deliver best products to the community. And in CE-IVD, there's a change from IVD diagnostics to IVD rules that puts more control on the manufacturing side.
Right. Thanks, Manoj. I think there are no other questions, and probably we could just move to the next section.
Thanks, Harry. So in section number 2, we largely talk about, I just want to give an introduction to the path in an R&D development and introduce some terminologies that is good to know. Okay. So let's say, suppose you are starting with a product development, it starts with defining the philosophy of the product. So for example, in our case, we'd say the product has to be point of care usable, data connectivity has to be there, it has to be portable, battery operated, and it should have stabilized reagents, because we don't want user to have a -20 freezer to keep the reagents, or a UPS for the entire lab to, so that if power goes on, reaction stops. So basically, these are the policy or the vision mission statement of the company that drives the product development around. So once you have an idea of the product philosophy, then we go into defining the product in detail, what we call it as target product profile. This is about an in-depth understanding of what the product is, where it will be used, who's my user, who the population going to use that, what are the costs involved, and what regulatory path we need to take, what clinical evaluations we need to do, and what is your competition, etc. It's about 40-50 parameters which define the product end to end. There's a lot of threadbare discussion going on to this, and this also becomes a central document that helps everybody in the team to understand, because when you have a product development, we will have team coming from multiple disciplines. You have engineers, you have biologists, chemists, and you have software. And this is kind of a central document that helps everybody to get an idea about what is being worked on. And there are a lot of target product profiles out there in the website. And you can, if anybody want to know what exactly goes into this, so you define something minimal requirement, optimal requirement. And you can look at this website here down called finddx.org TPPs. These are mostly on diagnostic TPPs, but this gives an idea of how to write a TPP and how to define a TPP for a product development. Another important aspect is called the technology readiness levels. So this is an important aspect in defining where are you today with respect to the development. So these numbers TRL 1, 2, 3 up to 9 indicate in which stage you are. So most of them, the concept or an idea originates in academy, in a fundamental research, and once they have an idea and then they think that a product can be idea can be made into product, they handhold with an industry, and there's a collaborative development period, and then the industry takes over from there. Okay, so this is a stage development. This basically originated from NASA and DoD, but it is largely utilized across the industry. This is one word answer. If I are TRL 3, now we have completed this much of development, we can move to TRL 4. And typically, industry starts at around TRL 6, primarily because the investment starts growing exponentially after that. It's like a huge investment, and you cannot have a look back once you start doing a product development in an industry. So academia developed the initial concept, we call it as a concept or call it as critical technology element, and then we have a collaborative development where you make it a prototypes and testings and some clinical evaluations. And then once that reaches a level and we have a discussion to decide on continue or not to continue, and to continue, then most of the work is taken over by the industry to commercialize and go on. So this gives a very good scale or platform to define what all need to be done towards product developments, and tell somebody, maybe your investor, I'm at a TRL3 or a 6, and it's a very short form to explain where do you stand now. Now, it's not just TRL, just be aware of a couple of more similar technology called manufacturing readiness level, customer readiness level. So what do these things mean? It is not like we have developed a technology, we transfer to the industry and they will manufacture. So it's not just like that. At early in the technology development, keep an eye on how we are going to manufacture that product or process. What are the available manufacturing technologies for that particular process? So for example, if you're taking a fluidic cartridge for diagnostics, it involves plastics. And what are the materials that is available to you? Because now we're talking about bulk purchase. Maybe you're purchasing in tons. And what are the materials available? What is the cost of each plastic? Are you working with polypropylene or polycarbonate or something else? What is the industrial process for assembly or manufacturing of that? And what machinery required? What kind of skill set required for people who run those machines? because all this becomes part of your costing. How the cost of the product depending on throughput and how many test kits you can create per minute or per hour in the assembly line that defines your manufacturing cost. So this goes in parallel to your technology development. It is not happening after your technology is developed. Let's just keep that in mind. Another technology or concept is customer readiness level. That is also runs in parallel. An evaluation of customer readiness. What does this mean? So this is to say that suppose I create a product, are my customers ready to use that? So let me give an example. Suppose you're talking 20 years back and you created a wonderful smartphone. At that time, internet is not so common or too expensive. So that product won't be accepted very well. So you just see if you switch off your internet connectivity in the smartphone, what can you do with that? So the product is there, but customers are not really ready to accept that. On the same line, RT-PCR today is quite well aware. People are aware of the usefulness and what it can do, what it cannot do. A lot of RT-PCR tests will come up now on various segments because customers are ready today compared to last 5 years back or 10 years back. So these three terminologies define how the technology is progressing and whether the manufacturing segment is ready to take up that and customers are ready to take it up that. And another important topic required is this is for students' information is what FMEA. FMEA stands for failure mode effect and analysis. Like whenever you're developing a technology or product, it is prone to failures in various stages and various conditions. So FMEA stands for identifying in what all mode a technology can fail or a product can fail. And if it failed, what is the severity or end result? Whether it creates a wrong result or it fails to give results. And what is the likelihood of occurrence of that failure? And last, in case something is failed, is your system capable of detecting that or will it pass on the result to the customer? So these three things, the severity, occurrence, and likelihood of the detection is evaluated by what you call the what if. So suppose you have made a technology and let's say you have made a reaction that is very optimal at pH 8. So we deliberately move the pH on either side, lower side or upper side, and see when the technology start to fail. Just giving an example. So, and we call it as guard banding. So does it have a tolerance of plus minus 1 pH or half a pH or 0.1? Then that defines the how severe is the failure. Just one example. So once you identify the severity or the chance and the detection method, we calculate the risk priority number. Risk priority number is a multiplier of occurrence, severity, and detection or detection ability. And based on this priority number, we work on eliminating these failure modes. And the end result would be these failure modes are eliminated. Or if you cannot eliminate it completely, the severity on the product is reduced, or the chance or occurrence of the that failure mode is reduced, or in worst case, even if it fails, the system knows that it is failed. For example, if you have done a PCR and the signal not amplified, you got a flat line instead of a growth curve, you call it as negative result, but that is not really true. You have two scenarios that can come there, but one is that okay, the sample was negative, so not amplified, or the PCR just failed. Something failed, the enzyme failed, and you didn't get an amplification. So how do you know that it's a true negative or it is something due to an aberration in the process itself? So what we do is that in every PCR, we add a component called internal positive control. That is a synthetic DNA added along with the sample, which goes through extractions and come to PCR, and that is amplified in the same reaction so that all your enzymes and reagents are working fine. So only when internal control amplifies and then your target PCR is negative, we call it as a negative. Otherwise, it is an invalid test. So that's just an example. How do we bring in diagnostics into the diagnostic machines to identify failures, etc. That's very important aspects, and this also help you in refining. If you are in research activity now, maybe in your PhD or master's, these things help you to refine the technology, especially if you're looking at a technology or a product development as part of your research. These things have higher value whenever you talk to an industry, like have you done an FMEA or not. Okay. And that's more about that. So for the students now, mostly what questions are asked, what kind of skill set you need now when you come to an industry for research, etc. So it is like it's more like a thought process walking through the maze. You would have done, I think people have done Balaraba or something, and you can why you can take a break, games in that. But you are not physically walking through this maze, but these are your thought processes channeling through the maze. And many times you get stuck at roadblocks where you don't know how to proceed, and your reactions or experiments are not going well, and you may have to make a return and come back and see where you have made a mistakes and take another path. So all these keep happening regularly. So I would say these 5 things are the most important skill sets that you need to develop if you want to pursue a research career. But first of all, you need to have a passion. It doesn't mean, don't mean which field, maybe biology or chemistry or drug design, etc. But you need to have a passion for that particular activity. You need to have good logical thinking and reasoning ability, because that help you to channel through this maze and go directly to the success path. And a lot of quantitative understanding of the technology. Not just understanding, but quantitative understanding means the magnitude and effect of a variable and how much that is going to affect the process. And most importantly, all these are done in a teamwork, in cross-functional team. CFT means you're going to work in a team involving multiple disciplines. You're working with chemists, biologists, electronics engineers, mechanical engineers, or software engineers. So effective communication between all these team, understanding their capability and limitations is an important fact when you're working in a product development environment. And most important is perseverance. Like often, and I would say most of the time, the reactions or the experiment doesn't yield in a success. Many times it fails. And you may have continuous failures for days or weeks, and we don't know what's happening. So we need to still hang on like a cliffhanger on a mountain, and then think again, analyze your results again and again, and then that's when I would say you put on a Sherlock hat on your head and then being investigative about the what is going on and how to get it right. And many of the patents or ideas come from that particular segments when you analyze your failure, because every failure is telling you something. You can either worry about that or you can take it positive and think about new ideas. In fact, one of our major product that we have in sample prep was a drastic failure 10 years back, and we couldn't work it out. Finally, we realized that that's a better way of sample processing than worrying about taking it as a failure. And now that is one of the globally most economic sample processing technique, and people across the world is looking at that. Just to give you some examples on that. So I just want to quickly run through our product, because having talked about technology development, so where do we stand with our product? Our product is largely known as TrueNat, and it's a point of care molecular diagnostic workflow. And last 1 year, we have deployed it all across India, more than 3,000 devices installed and running. Most of them running for COVID and many running for TB and other diagnostics. And we are there from in every state and every union territory, from either Lakshadweep to Andaman to various parts. And how this works. Basically, this is the package that we offer. It's a portable diagnostic device. It involves a sample processing unit, a real-time PCR, and some printers, accessories. And it's a lightweight components, about 1 to 2 kgs, portable, rugged, no AC, air conditioning required in your lab, can run up to 40 degrees, rechargeable battery powered, so you don't worry about power cuts. You don't need a UPS. It can run the whole 8 hours without the power. So typically people can use it in the daytime and plug in and charge overnight. And it all comes with ready use sterilized and -20. So these are all stable in room temperature conditions. And proudly made in India, because the entire technology, every segment of this is made in India and manufactured in India. And now this is becoming a global product out there. Now, to give a summary of what exactly is this product, the heart of this process is a microchip. So here, if you look at the white portion of the chip, this is a thermal cycler. You would have seen thermal cycling. Basically, this is a portion or a chip that heats and cools fundamentally. It heats to 60 degree and then will go to 95 degree, come back to 60 because it has an embedded circuitry in that. This is a ceramic chip. And other part of the chip has the electrical connectivity and also have a memory. It's a flash memory. Basically, because when you're giving a test for point of care, like in a primary healthcare, you cannot expect the same skill set in the user as in a centralized lab. So we need to build in a lot of self-checks and diagnostics to the reaction so that, for example, if they have taken a dengue chip and then trying to run malaria, it cannot run, you should not run that. So when you put a chip inside the system, it knows what it is meant for and what disease it can detect by itself. So it doesn't allow user to create a mistake. So like that, a lot of self-checks are done in the system that enables us to deliver these chips to the remotest place in India and lower more skill set. A microscopy technician can start running PCR without even knowing what is PCR. So that is the level of engineering that got into these developments. And also, if you'd seen earlier, the standard curve that is required to convert a CT to the viral copy number, that is also fed into each and every chip. And in every chip, we do triplex PCR. A triplex PCR means we have 3 reactions happening in parallel in the same chip. One is your internal control, and the other 2 are targets. For example, we can do a PCR of dengue and chikungunya in the same chip. So you know that the symptoms are overlapping, and maybe doctor may want to know which is which. So instead of doing 2 tests, you can do 1 reactions and identify whether it is dengue or chikungunya, and if it is dengue, how many virus per ml of blood sample in the patients. So and the systems are built around these chips. So let's say we're starting with the blood sample, we have a device that processes the RNA or DNA out. Right? It basically works on a fluidic cartridge. You load the sample along with 1 reagent and insert into the device and press start. That's all you need to do. And everything happens within the cartridge on DNA, the lysing of the bacteria or virus, purifying the RNA or DNA, and you get the DNA at the end of about 20 minutes. So you can load in the machine, walk away, and come back after about 15-20 minutes. You have your RNA ready. So once you have the RNA ready, it goes to the second machine, which is called an analyzer or to lab, wherein we insert this chip into the slot and you select what you want to run on the screen, and then it runs the PCR in this machine and shows the graph on the screen. It also connected. Nowadays, we talk about data connectivity a lot because all these machines have Wi-Fi, Bluetooth, or 3G or 4G data enabled. What you can do is that after every reaction, the data from the system can be pushed to a cloud and you can analyze it in one location. So for example, you have 3,000 devices in India, all across India, and they're running COVID test, and we can monitor, or a central ministry or healthcare authority can monitor how many tests are being done now, how many are positive, and which part has more positives, which part has more negative. I'm just saying some examples. So nowadays it's all about data and connectivity, and we don't need somebody to write down this data, make a report, and send it to the higher authority, and then collect all this and then analyze. All this happens automatically if you need. So these capabilities are built into the systems, and it is future ready. We can say it's an Internet of Things, and we can do a lot of real-time analytics based on the system. And on the menu, we have a large number of tests, about 20 to 30 tests right now in the market. And we can look at all these are some kind of infectious diseases. One genetic test here called HLA-B27. So one chip, but we can do a lot of different diagnostics based on what reagents is loaded on these chips. And I don't know how many of you are aware of this recent outbreak of Nipah in Calicut, and we got an emergency use authorization of for using this test for Nipah, and that is our last test added to this menu. Okay. So what is our path? So if you look at the development path started in 2006 through a Government of India funding through CSIR, Nimidly project, where we ideated a concept to a PCR on a chip, and the chip is very different at that time, the prototypes. So we had a working prototype by in 2 years, which was just doing one PCR at a time. which is hepatitis B. Then we realized that, okay, we need to add an internal control. We made into Duplex PCR in 2010. And in 2012, we formed the manufacturing activity and formed Molbio for manufacturing. This is in Goa. And the product was released next year. And we have got the ISO clearance of CVIVD. And because it's originated as a government project, it was launched by then President of India, Pranab Mukherjee, in a National Science Day. So beyond that, the product further evolved to what is running now. It's called a Triplex Pcr in 2016. And 2012 to 2020 was long evaluation of clinical trials within India, outside India, on multiple countries, leading to WHO approval for the TB diagnostics. Nowadays, we are part of the National TB Control, National TB Elimination Program. You would have seen all these smear microscopy centers now getting converted into molecular test. So when we go for a TB test, it was done under by microscopy. Now that is getting converted into molecular diagnostics. So you have a precise diagnostic of TB and also a rifampicin test as a follow-on test to identify whether it is first-line treatment resistance or not. So that is our, in a nutshell, how the products is evolved and how the technology is developed and how we are in the market now. And post 2020 is a whole different bar game. And COVID started in March, and the entire focus was to use these products for COVID in various places. And last 1.5 years, we have scaled up our manufacturing tremendously from few thousand tests per day to nearly 1 lakh tests per day is our current installed capacity. And now we are going globally with all these things out. So Harry, I think I'm just run through quickly. I think the time is running out. So maybe I done quickly through this. That's about the overview of technology development. And I have one case study at the end for the participants. Anything to discuss now?
Yeah, but actually, before you go ahead, Manoj, amazing. This last, the last session which you did, it really was amazing. And I realize one thing now. So actually, for the people who are listening in, this session is very different from, the sessions that you would have heard in the past, because this actually talks about products, unlike the rest of, the sessions which you would be hearing or the kind of work which you would be mostly associated with. So if you have a dream of becoming an entrepreneur, okay, if you want to start a company of your own, I would really urge you to go back and listen to the session. It will be on YouTube very shortly. Please do go back and check this out, because you will get a lot of insights into it. Manoj, adiboli, in such a short span of time, you were able to cover so much of terrain. വളരെ simple ആയിട്ട് പറഞ്ഞു കഴിഞ്ഞാൽ 3 different aspects. See, for a product to succeed in the market, one, the technology has to work, right? I mean, you talked about technology readiness level, right? The second thing is that you need to be able to produce it at a level where you actually make money out of it, right? I mean, if your manufacturing cost is too high, you will not be able to make money out of it. So that's basically the manufacturing readiness level. And finally, you do all of this, somebody has to buy it. That's the customer readiness level. Idu moonu illengile, your product just will not succeed in the market. So any budding entrepreneurs out there would really suggest that you go back and check this session later on once it is put up in the YouTube. Now, there are quite a few points which have come up in the chat window. Most of them deal, appreciate your session. There was an input which came in about, the curriculum changes which is required because the current B Pharma, M Pharma curriculum, etc., doesn't cover some of these industry aspects. So probably that needs to come in. And there was another question which came in, which referred to the fact that a GATE or a NEET kind of exam has got more engineering concepts. And since, the B Pharm students are more focused on the medical side of things, how would you prepare for an exam? So that was one question. And maybe if you could just take that, and after that, we can move on to section 3.
Yeah, thanks, Kari. So definitely when you're looking at a curriculum related thing or a competitive exams, so see, I will tell you frankly, I have not prepared anything for my GATE exam other than studying what is taught in B Pharm. So I didn't even plan for a GATE exam till my final year or the exams are there. So it is more about better understanding your BPharm curriculum itself is so diverse. It has elements of engineering in that. It's have biochemistry, biology, chemistry, and these are all fundamentals. It's about choosing your area of interest, and I'll come to this on the last slide. But it is not just about learning, remembering, and reproducing it in your exam. It's about higher order thinking skills. How do you gain more information beyond what you are taught? Because when we were studying, the information source is either what the teacher is teaching you and few books in the library. Other than that, we never had an access because there was no internet at that time. There were no computers at that time. So we were limited with these informations, and remaining is in how many books you go and read in a library. But that is not the case today. If you want to learn on something, everything is on your fingertip on the internet, and courses are there, content is there, information is there. It's all about identifying what you want to do and never get satisfied with learning something. It is about learning more and more, moving more deep into that, and that will eventually fetch you in the competitive exam, because competitive exams are about applied questions. It's not just asking you to reproduce what you learned in the direct way. It's mostly indirect way of questioning you to understand what is your level of understanding on the topic. So it's not about learning something new. I would say learning things in more depth that we can. It not just for competitive exam. That is very much required for you to be in a career. Getting into career is one side where you studied, but surviving in a career is much more a difficult task. Okay. So and all this needs an in-depth understanding. So my case study coming in next 2 slides is a little bit on this.
Yeah, before that, moving to the section 3, shall I launch a poll?
Yes, Prashant.
Yeah, go ahead, Prashant.
So I'll just keep these slides alive if anybody want to look at that while you answer the poll. So let Prashant, I think let the poll happens, because if time is running out, I will take this these things and okay, fine. So in this slide, basically, I want to talk about a very emerging technology called next generation sequencing. I think you would have seen this as in a great scale. All the COVID genomes and mutations or classes coming from, if I said 51 lakh genome out there, that's a phenomenal number to appear in last 1.5 years. No other diseases is so well studied. So where is all this information coming from? You take a sample from a positive patient, you subject through a analysis procedure called sequencing, and then you have the whole virus genome. So one technology out there, this originated in Imperial College London, and then this is co-developed with a company called DNA Electronics. Is called Ion Torrent. Ion Torrent basically has a chip, which if you put the sample in and run the chip, it gets the sequence out. Basically what it happens is that it amplifies, before uploading into the thing, you amplify the DNA or by PCR. Then all the amplicons are bound to some nanoparticle, and then we run the PCR again on that strands. So let's look more in depth into this thing, what exactly happening at molecular level. This is called sequencing by synthesis, or SBS in short. Let's say you have a strand where you don't know the sequence. You add one nucleotide at a time and then see whether it is incorporated or not. For example, let's say it says already incorporated 4. The 5th one, if I add a T, because A is here, a T will pair come perfectly here and the reaction will happen. Now, if the reaction happens, there is 2 byproducts formed. One is a H plus ion and another is a pyrophosphate. So how do we know that this reaction has happened or not? So the way is that not by looking into this, look at this. If an H plus ion is being produced, you know the reaction has happened. So how do you know H plus ion is being produced? Just look at the pH, right? If an H plus ion is being produced in a reaction or in a buffer, the pH will drop. Okay, and if no pH drop, no reaction has happened. So basically, this is the setup, what is there in this chip in a zoomed up level. So we have bead with all these DNA strands, and then you flow onto that A, then T, then G sequentially, and in between check whether the pH is changed or not. So for example, let's say if you flow A, check the pH is changed or not. Do not change, wash it out, then send T, change or not, then send G, then change it or not. Maybe when you send C, then you will see a change. And this cycle keep repeating for every base, and essentially you will see a graph like this, like that. When I put in T, I got a signal. When I put in A, I didn't get a signal. C, I got a signal. G got a signal. So G and T never had a signal. A and C gave a signal. So if you read all these things and also look at the magnitude of the change, this is the first one is double of the second one. So this probably indicated two T's together, T, T. So if I would have to read out the sequence, I would say the unknown sequence is T, T, C, A, C, T, C, G, A, C, T. I hope you got that. So this is how we do a sequence readout in a reactions. So I don't want you to go into the details of that. I only want you to look at how simple is the concept. We are just monitoring a change in the pH, and you are effectively getting a DNA sequence out. So if you look at many of these fantastic technologies out there, it's a very fundamental concept which you would have studied. I think every BPharm student would have studied a pH change and how to pH is negative log of H plus ion concentration, Henderson-Hasselbalch equation, blah blah. But this becomes a concept to a great product in the market. Okay, so this happens not only here. Almost every product in the market are based on very fundamental concepts in science, and it is how cleverly you use that and how cleverly you design that, and then how you formulate into a cheap or consumable, and you create a machinery out of that. Now, if you understood the fundamental concepts, you just need to know that every DNA correct incorporation produces an H plus ion, and if is a very important. If you can measure that pH, you can detect the reaction is happening and you can get the sequence out. Now let's just imagine there's a virtual scenario for the students. Like suppose if you are part of this development team, so you are a student now, imagine several years later, you got into a job and you are part of the team who's developing this machine out there. It's a virtual scenario. We can imagine, everybody can imagine anything. Let's imagine that virtual case. So I put that case here, like if you are in a cross-functional team. So what do you mean by cross-functional team? I keep you here in the middle. I call a BPharm student or as a chemist because you have more of chemistry than biology, and you study a lot of chemistry out there. So let's example you are a chemist in the team. You have a biologist, a partner with you, a colleague. You have an electronics engineer with you, and you may have a design engineer, we call it a microfluidics expert in that team, and you may have your boss. Okay, of course they're in the team. No need to ask. Now you need to interact with all these team members. Let's say in the discussion, you are in the first meeting about conceptualizing this product, and all of them have done their homework and they come up with what they can do. And the first person is telling that, okay, I can do a PCR at around pH 8 to 9, and I can get around 10 power 9 DNA from starting from one copy. So that is what he creates. So, and he creates a single stranded DNA because that is what is required for sequencing. So he says this is what he can do. And similarly, the microfluidics fellow may say, because that's his job, to create a cassette and then pass all these ATGC one by one through a nano droplet. He will talk about nanoliter volume. Because why nanoliter? Because all these are expensive chemical. We cannot do the reactions in milliliter because it's quite expensive. And for another reason that the pH change will not happen if you do it in a bulk volume. So it's better to do that in a very small droplets, essentially in nanoliter. So he says, okay, let's say I can have a plastic cartridge and all the driving circuits inside that, and I can do the reactions in 100 nanoliter droplet volume. Okay, that's what his capability is. And the electronics engineer says, okay, I can make a sensor. If you look at the previous slide, it's not a pH meter. It's not a glass electrode that you put in, because you cannot put a glass electrode into nanoliter volume. You need a then another kind of sensor, which is called a FET or FET electrode. And he says, okay, I can make a sensor very sensitive down to change of 0.02 change in pH. So that is 8.00 to 8.02, very sensitive. Now, all of them are telling the best what they can do. And maybe your boss now asks you, can you check the feasibility? Like, given all these conditions, all this scenario, will this product work? So that is a question to you if you are in a team, and you have to answer with explanations. And if you're not clear about all these questions, let me put that problem statement here. The question number 1 or first level question is, if you have 10 power 9 H plus ion being produced in a droplet of 100 nanoliter, will that cause a change in pH of 0.02 at least? If all these things can happen, the technology will work. Okay, because all of these are not your bulk reaction. These are going to be in a very small volume and very, very minute change in pH. It sounds very minute, but given all these conditions as input, will it even work? That is your first level of questions. Now, let's say you have found the answer for that. The next question for you would be, the biologist says he can do the reaction at a particular pH range. So you need a buffer there. So if you see, these two are opposing things. If you use a strong buffer, you can buffer well, but it will not cause a pH change. But if you use a weak buffer, it will cause a pH change, but you may not be able to do the reaction in that. So given a choice of different buffers, which buffer will you use? Like, is it a tris buffer or phosphate buffer, or any other choice in that? Okay, you can work out that too. And even if you find an answer for question number 2, question number 3 is, what molarity can I use in that buffer? Like, if I use a strong molar buffer, yes, I can buffer very well, but it will not have any change in pH. It will nullify. So what is the maximum molarity of the buffer that I can use there? All these are your calculations that you need to do. And now see, this is not a exam question so that you have to remember something from your mind and you have only a bunch of paper and pen and calculator with you. This is an open textbook problem. You can refer anything. You can browse internet. You can talk to people. You can look at literature. You can go back and study. You can do anything. You're not limited by anything like in a classical exam, but you need to find an answer with evidence, answer with numbers on that. And that is a kind of a typical scenario that you may encounter when you get into this kind of a development team, because it is not, say, I would say that you would have studied all these things much earlier in your curriculum. It's not like, or even your +1, +2 curriculum, but applying that to a practical scenario is a little bit different. It's not just remembering some equations and saying that, okay, this is what it is, but it is about doing the calculation, doing the math yourself, and then seeing whether it is possible or not. And that shows a little more. Maybe you had studied again to do that, and you can take your own sweet time. And then I'm not asking an answer right now. Whenever you're done with that, you can post either through email or the WhatsApp group back to us. Let's see how well you work out. You can use any tool, like you can use Excel or whatever you want to use in that. And to make your life a little more complicated, I would say scenario number 2, like, given all these are ideal cases, as the research going on, you cannot trust all these people around you completely. Like, they have promised something, but when they start their work, they say, okay, I tried my best to achieve 10 power 9, but I'm not getting really there. I can get to 10 power 8 only. Will it still work? Or this fellow say that I had tried my best in the plastic, but I'm not able to make 100 nanoliter, but maybe 200 are the lowest volume. Or even this fellow may say that I tried for 0.02, but due to the noise, we need to settle around 0.03 or 0.04. None of that makes your life much more miserable, not just a direct calculation. Now you have to factor in all these possibilities of failure, and you still need to answer your boss. Okay. And that makes calculations more iterative, considering all these failure scenarios. So now we started doing FMEA on that concept, and you started understanding in what all conditions my technology can fail, and what is my limit of performance, etc., etc. So that's it. That's just a quick virtual scenario I want to throw at you. take your time, and if you're interested to solve these things, let's see if you can solve, and then we can push back that answers. So maybe I would request you one page write up on these things and see how you have solved these things and get back to us. So with that, I think I would come to the end of the session, and I would now leave it open for Q&A with this last slide. And thanks for everybody to listen to this webinar on a Sunday evening. I think the match is coming up. It's raining, but I hope you have some useful time spending with us on the webinar today. And this is image on the right. It looks more like a technology readiness level, but that's not TRL. It's about your learning level. It's called Bloom's taxonomy. It can go to various universities. They use that to evaluate the students' learning level. So it's about the lowermost level where you start with, okay, you're studying something, you are able to remember that and then reproduce back in an exam where the question is very direct. Or you have a little more understanding and you're able to explain maybe an out of syllabus question or something like that. Or you're able to apply that concept into something like a small technology, or you're able to analyze and create ideas on that, or even further, you're able to make a stand or justify on a technology, or even create a technology, something new out of your learning. So this is the pyramid of learning, and it's always continuous, and the level goes more deeper and deeper as you go up on that, and then that's a self evaluation of whether I have really learned something or really understood, and did I realize the real concept behind that. So if you want to ask yourself, this is a fantastic concept made in various universities, and they follow all these things. Hari, I think I come to the end of the session with that, and hope it was good enough.
Thanks a lot, Manoj. I don't see any other questions. And again, the last session, the third session which you did, again was phenomenal. For the simple reason that, I mean, it throws light on a facet of your industry, which is not really covered in the regular pharma world. In the sense that it is all about teamwork. Okay, it is, in some sense, it is a Virat Kohli problem. I mean, you might be the best batsman in the world, but you need to find the strengths of the other people in the team and get everybody to work together. Only then can you win. And you need to know what really is the true strength. Are they performing to their potential, and how everybody comes together, right? So that's a fascinating topic. And again, for anybody who is interested in starting their own company, joining into a product world, okay, or doing research in a bigger company, the teamwork aspects of it, what you do has to fit in with what everybody else around you is doing. That's something which Manoj had just touched upon, and that is really, really interesting. And before we wind up and before we thank Manoj, I'll just take a quick look at the poll which we have conducted. And the first question was about, do you wish to know more about, what is it that you wish to know more about? So the majority of the team, majority of the people who responded felt that pharmacogenetics, almost 53% responded with pharmacogenetics. And the rest is a mix with, 30% talking about tech details in RT-PCR, 27% about companion diagnostics, 27% about diagnostic regulatory tools, etc. So it seems that, the majority of the people who responded seem to be interested in knowing more about pharmacogenetics.
Yeah, this is natural.
Yeah.
Having our audience on pharma, I would expect the same thing. So,
right. The second question was an interesting question. It was about, have you used any software tools? And this we kind of, got responses all over the place. I mean, there was 17% for coding software, 17% for data analysis and visualization, 33% for computational biology and drug design, 43% was for any other. So which essentially means that, I mean, it is a very, very fragmented understanding of the tools which are available in the market, and which again, I guess, points to one of the points which came up in the chat window as well, the need to include some of these aspects also into the BPharm and MPharm curriculum so that people are more familiar and more aware of these tools. The last question was about, would you like to know more about TPP, TRL, MRL, FMEA? Overwhelming one was all of the above. 77% said that, they wanted to know all of the above. So I think you really achieved your target,
Thanks.
which is about
Good to know.
which is about making people want to understand the product world more, the diagnostics world more. And I think there's a lot of interest in the audience, and feel free to kind of reach out back to Manoj with the response to the case study that he had suggested, as well as any other doubts.
Hari, I really had a doubt before I gave the webinar or preparing for, because this is little away from the main curriculum, how people will take it up, and I was thinking back and forth on how to present the diagnostic into a pharma audience, but I hope I could do some bit of knowledge sharing here
Absolutely.
and there.
Absolutely. Yeah, right. Thanks. And with that, I'll pass it back to Ranjish. Thanks a lot, Manoj.
Rudraksh, does it have
Thank
any comments?
Yeah, thank you, Manoj. I was just waiting for Harry to finish, and this has been so meaningful. Just the effort you have put into this, the impact it has made, at least based on all the responses and the feedback, and just the quality of the presentation. It's so meaningful. I think if I were a student today, I would just go back and listen to this, like Harry said before, back into the YouTube, listen to this. There's so much here, and I reach out to us, and I think as we go through this will set a wave, Ranjish also introduced with Pandit Nehru, that we are setting up the next generation, and it's the small part we are all playing. We are very glad that we had a good participation. So thanks, Manoj.
Thanks, Rudraksh.
So shall we?
Ranjish, over to you. Yeah.
Okay, I invite Dilip sir for offering the vote of thanks. Dilip sir.
Yes. Yeah, it's a great day after all. Dear all members of the CoPS Global and all delegates, it's good to have everyone connected once again for a great course. It is the Children's Day today, and it's the third talk in our webinar series. The molecular diagnostics is an advanced field of science and a relevant one now. But our dear Manoj M N, the man behind the TrueNat, has made it very simple and understandable. I really hope this session will make some sparks in the minds of our new generation, which is our target group. On behalf of CoPS Global, I would like to express our sincere gratitude to Dr. Manoj M N for the wonderful presentation. Next, I would like to thank the moderators, Rudraksh Sharan and Mr. Harry Jose, for their valuable contribution to this presentation. And I take this opportunity to express our sincere thanks to all the delegates participated in this program on behalf of CoPS Global. Thank you all.
Thank you.
So with that, we are winding up. So see you next time. We'll be back again with some other topic.
Yeah.
Thank you.
The feedback form is given in the chat box. You can fill it for getting the e-certificate. Participants.
Okay, participants, please fill in the feedback form so that your participation certificate will be mailed to your email ID. So that's it. Thank you. Good night.
Thank you, everyone. Thanks for joining.
Thank you.
Thank you. Good night.